set? Okay, we're going to go ahead and get started. I'm Dr. Hines. I'm the Nuclear Engineering Department head here at the University of Tennessee, and welcome to our Nuclear Engineering Colloquium program. Today, we're very lucky to have an assistant professor here from Clemson, Dr. Nicole Martinez. She's within a unique department that addresses broad environmental issues associated with anthropogenic. I had to look that up. Although Siri says it's pogenic, not pogenic, but Siri doesn't always. She may not be. That may not be her first language, I'm not sure. Maybe assembly is her first language. And natural radioactivity. So since one of them was that and the other is natural, I figured that must be man-made. So that's human-made. So anthropogenic, human-made. That's as much as I'm going to teach you today. But she received both her master's and her PhD in radiological health sciences from Colorado State, specializing in health physics, radioecology, respectively. Prior to attending graduate school, she served in the U.S. Navy as a nuclear power instructor and radiation health officer. Her current research interests focus on the behavior and effects of radiological contaminants in the environment, to include radiation transport modeling, improved dosimetric methods, low-dose response in microorganisms, the influence of plants on radionuclide mobility. In addition, she's interested in non-technical issues within radiation protection, such as ethics and diversity. She has a long list of honors. Let me read two of them. She's the recipient of the 2018 Bo Lindell Medal from the ICRP and the 2019 Elda Anderson Award from Health Physics Society. So welcome, Nicole.
Thank you. So can you hear me, web person? Great. Can the rest of you hear me okay? Great. All right, so thank you so much for having me. When Dr. Hines invited me, he said that he's interested, and the program is interested in nuclear engineering from an environmental management standpoint and kind of broadening connections and networking in that area. So I've chosen a topic today that's relevant to environmental management from a nuclear perspective. But before I get into my specific talk, I'd like to give a little bit of pitch for Clemson University. We are number 24, top national public university, according to U.S. News and World Report, but number 1 in universities that students love. So we're top of student happiness, which is really what matters, right? Part of that might have something to do with the fact that we've been so good at football recently. We've been to the national championship about 4 times in the past 5 or so years, and we won 2 of them, not this past year. But nonetheless, that's this picture on the left. Notice these balloons. We don't do that anymore. The environmental department helped stop releasing balloons. That's not good for the environment. So we finally persuaded the football program that that's correct. So now we have only the fireworks instead. But still very exciting for Clemson football. Just as big of fans as the University of Tennessee. It's funny, I was mentioning to some of my colleagues earlier, I'm actually from Tennessee. I was born in Maryville, even though I grew up in Texas, and my parents have since retired to Maryville, and they both actually went to graduate school at the University of Tennessee. This is my first time on campus, so it's my pleasure to be here today. Clemson's in a much smaller town. It's very interesting to me to get to Knoxville in this big city and then surprise the city turns into university, whereas Clemson is a very small town. Like, pretty much the city is the students and the people who work at the university. It's true. It is true. All right, just a little bit of history of the nuclear program. My department is environmental engineering and earth sciences, and within that we have a lot of variety. We have environmental engineering, we have biosystems engineering, we have geology. In the undergraduate programs, we also have a nuclear minor as an undergraduate program, but for a major, our nuclear is only at the graduate level, and we call that the Nuclear Environmental Engineering and Science program. Our health physics program is ABET accredited, and we also have an environmental radiochemistry program, which is predominantly a PhD program. But you can see we got our start in 1980. And then Tim Duvall joined in 1995, and some of you may have, particularly faculty, may have collaborated with him in the past. We hired a radiochemist in 2008. I say we, but that wasn't me at the time. I wasn't around at the time. You can see Brian is in the field there. Lindsay Schuler-Nichols was hired in 2011. She's a material science engineer, does a lot of mineralogy, a lot of work with nuclear waste forms. And then I was hired in 2014. I'm a health physicist, but also a radioecologist. So they were looking for someone on the biosphere side of things as opposed to the what's going on fate and transport-wise. They wanted a food chain transport and potentially effects as well. And then 9 graduate course offerings as of 2017, and then 2015 is when we first got our minor. And we've jumped up. We started out with just a couple people in the minor, and now we're like a dozen. So for a pretty new minor, we're doing pretty good as far as that goes. We also have a center. So the Nuclear Environmental Engineering and Science is our graduate program, but we also have a Center for Nuclear Environmental Engineering, NETREM, as we call it. It's a long acronym that needs some work. We need a public relations officer to handle that. But we have not just people from environmental engineering, we also have people from chemical engineering, from plant sciences, from environmental toxicology. All these people working together, interested in nuclear issues, come together in this center. And so really that's in investigating nuclear technologies and also naturally occurring environmental radioactivity for the purpose of risk assessment and also for environmental stewardship. One of our focuses, relatedly, is also nuclear forensics, although that's a little bit tangential to what we're going to talk about today. So our dominant programs, environmental health physics, radioecology, and environmental radiochemistry. But a collaboration with the University of Tennessee would be great to get that extra nuclear engineering aspect in. Acknowledgement. So the work I'm talking about today, I didn't do myself. I'm hardly in the lab anymore. So the person largely responsible for this is Dawn Montgomery. She's my PhD student. She should be graduating this year, finishing up her last paper. So if anyone has a job opening, she is my rock star. We'll be sad to see her go. The principal investigator is Dr. Brian Powell, our radiochemist. He's here with a quasi-portable HPGE on top of a plastic pipe and monazite outcropping local to us. And then Dr. Thorell and his PhD student, Nimisha, are in plant sciences, and so they helped us with some of the plant physiology aspects of this project. I also have to give a shout out to 3 undergraduates, particularly Scott and Hayden. You'll see why in a minute. I'll tell you what they're responsible for. And this is funded by EPSCoR. The overall project, the big picture, is called Radionuclide Waste Disposal, Development of Multi-Scale Experimental and Modeling Capabilities. So really we're just trying to identify all the interactions that control mobility of particularly risk-driving radionuclides. We figured out what risk-driving radionuclides are from the previous experience at Savannah River Site. And this work is specifically for the purpose of environmental management and responsible handling of waste at the Savannah River. And we want to be able to identify these mechanisms, link them together, and scale up, so that eventually we can go from a laboratory experiment all the way up to a field scale experiment, and to do that reasonably accurately. So that's why we're interested in all these little steps. Particularly neptunium, technetium, cesium, uranium, and iodine are the radionuclides that we're interested in. Part of this project was also looking at different waste forms and how radionuclides might disperse from those waste forms, delineating different processes, and then not just spatial scales, but also temporal or time scales as well. So what that looks like, we have our repository per se, and we can have radionuclides mobilizing from there. So we take different types of waste forms with radioactivity in them, whether that's cement, whether that's ceramics, and we're housing them in a lysimeter facility. So these are like columns, I don't know, maybe a foot in diameter, filled with dirt, and in the middle of it, there's a puck of some kind, I don't know, about the size of your fist, to represent this particular waste form. And there's various instrumentation in these lysimeters to collect a variety of different data to figure out how those radionuclides are moving. Where I'm involved is the biogenic portion, where we have a root, and it is figuring out how to get nutrients. And to do that, it might mobilize those nutrients and accidentally or inadvertently mobilize radionuclides, which then can influence if they go down into the groundwater, if they come up into the plant and into the food chain. And then for the last part of the project, we're hoping to model across those different scales. On the left-hand side of your screen, the blue Venn diagram, you can see some of the experimental work that's been done in this project so far. So on the left-hand side with biogenic ligands, there are plant folks like myself. And this bottom left is light imaging. This is a very thin slab with dirt in it, thin enough that you can shine just a regular light through it and get an image for preferential flow. We have some column experiments at the top looking at ion interactions, and then also redox processes. And this puck, this is a cement puck, and you can see the different coloration is oxidation. And then some other imaging techniques, electron microscopy, for example. Oh, and then this is microfluidics, this top picture here. Also, again, even smaller scale than our lysimeters. So that's the overall big picture of what's going on. And then this portion is just basically the plant portion. So we're taking an integrative approach for radiological protection of the environment. We want to know the plant's influence on biogeochemistry. I'm interested in plant uptake. And then extra, aside from this particular project, I'm interested as a radioecologist in non-human biota dosimetry. So plants and animals that are not humans, how do we figure out what their radiation dose is so we can adequately get protection of the environment? We scaled across different levels. So the first experiment was just batch sorption. Basically, you're just taking soil and water, and then we add some model ligands, so oxalate and citrate in different combinations, and get representative baseline KDs. From there, we do a hydroponic experiment. So the plant is in a nutrient solution, not soil. It's not as accurate or realistic, of course, as soil, but we have a much higher control over what's going on around the roots than you would in soil. From the hydroponic work, we also developed dosimetric models, as are pictured here. 2 different ones, which I'll talk about. And then finally, the experiment that's being finished up as we speak is the plant soil column. So we have a column of soil, 2 different ports here for injecting the radionuclide and also sampling pore water, and then we have a plant in there. So the idea is, after she finishes all of these experiments, she's going to try to model and scale all the way from using the KD and the data we got from the hydroponics experiment and see if we can figure out how to predict what's going on in the plant soil columns. So today I'm going to talk about the hydroponics, the plant soil columns, and then we'll also talk a little bit about dosimetry. Why do we care? One of the things is that it's consistent. So similar studies have been done, but with different analytes, different types of soil, and there's not a reliable consistency between the different types. So by using Savannah River Site soil in the KD experiment, all the way to the soil column experiment, we can be reasonably assured that that's, when we're trying to extrapolate, that's a reasonable extrapolation to even be trying to make. Similarly, with the plant species, it's the same plant in the hydroponics and in the soil column studies, and rather than use a model species like, say, Arabidopsis thaliana, we're using a local grass, so a grass that we have all over the place and that's likely to be found at Savannah River Site, making that a realistic approximation for plant uptake. We're also linking between experimental and computational dosimetric modeling. A lot of the dose models that are out there for non-humans are hypothetical and approximate because there's just not a whole lot of data out there on non-humans, specifically with respect to the environment, at least not good dosimetric data. And the modern philosophy of the International Commission on Radiological Protection is when we have radiological protection, we're protecting humans and the environment, and we want our protection strategies to take all of that into consideration. And these non-human biota dosimetric models are fairly novel. There's a couple fancy models that have been developed previously. none have been made for plants. And so there is an international effort. Recently, these fancy dosimetric models have been adopted by the ICRP for humans. And it's likely that we might eventually get to adopting them for non-human biota as well, at least for certain representative organisms, or for following a graded approach to screening for protection. If you do your conservative approach and you're not quite to the benchmark that you want to be at, then you can start adding realism. That's where that probably comes into relevance. The analytes that we are considering are relevant for legacy waste, special nuclear facility repositories, spent nuclear fuel repositories, and reprocessing streams. In particular, we are, of course, interested, being in Clemson, South Carolina, which is in the upstate, Savannah River Site, which is just down the road. That's what we're interested in. But of course, we have Oak Ridge. That's not much farther from us, and pretty much your guys' next door neighbor. All of these analytes have the potential to be mobile, long-lived, and hazardous to human health and the environment. The particular 4 uranium, cesium, neptunium, and technetium chosen in this project have a broad range of biogeochemistry, different sorption mechanisms, redox activity, solubility, etc. And then for your information, I have here the relative complexation and sorption affinity. So this is why we chose those particular radionuclides. In terms of the plant, I mentioned Andropogon virginicus. Here's that grass. You probably will see it on the side of the road. Broom sedge, it's a bunch grass, common in the southeast United States. The thing I didn't mention is you can kill it, but it's harder to kill it. So for someone like my student who's not a plant person, hasn't grown plants before, if the environmental conditions aren't ideal, it doesn't instantly die. I mean, she has killed some before. But it is more tolerant to stressors. So if we need to stress it for whatever reason, we can do that. Root exudates are compounds released into the environment, as pictured here. There are, of course, also microbial exudates that contribute also to organic acids, amino acids, various things like that being formed that can form complexes in the rhizosphere and other areas outside of the rhizosphere surrounding the roots. The plants do this to look for nutrients. A plant can't, walk down the street to a restaurant to get its nutrients. So if it doesn't have available nutrients, it'll send out phytosiderophores and try to solubilize iron or whatever else, and then use evapotranspiration to draw the water with the soluble nutrient up into the roots and to the plant. So the plant's not picky, or the chemistry is not picky about what solubilize. So there's the potential to mobilize heavy metals that aren't nutrients as well. Of course, that includes radionuclides. Having a nutrient analog is almost certainly going to increase the potential for uptake into the plant, so into the roots. Translocation means going from the roots up into the green part of the plant into the shoots. So cesium, potassium is a nutrient analog, which is an essential nutrient, and then pertechnetate has a variety of potential nutrient analogs. And the picture on the right-hand side of your screen is just different pathways for different types of nutrients into a plant. So there's a lot of different ways for radionuclides to get into our plant. Metal ions can come into the roots as either a complex, or they can actually just kind of drift across, right? There's nothing preventing them from going into the roots. Once they're there, generally they move with water, and they might move up into the shoot. At this point, the plant can recognize whether, usually, not always, but for the most part, the plant can recognize, yes, this is potassium, this is what I need to do whatever process, or, ooh, this is cesium, this is not potassium, it's not working for what I need, and then it'll try to either push it kind of to the older edges of the leaves, and in some cases, for some metals, the plant is smart enough to press that back down into the roots, which is pretty cool that plants have that strategy for responding to heavy metal stress. For the non-human biota dosimetry, we want to be able to demonstrate that our environment is adequately protected. For the environment we live in now and also for future generations, we want to make sure our actions are sustainable. And to really get a good protection strategy, we need a strong dose effect curve. And we don't have great dose effect curves for non-human biota. They're pretty much just wide bars of this is the LD50 for death of a certain taxonomic group. But really, there's more to it than that in assessing population level effects. The first step for us is figuring out good ways to determine dose. So if you think about it, I like to use birds as an example. If you have whatever meter you have, you stick it in your forest. You want to know what the dose to your birds are. And then you catch a bird, and you give that bird a dose that your meter says. Is that right? I see several heads shaking no. I'll say no for the people who are listening. No, because that bird wasn't like living on that detector, right? Maybe that bird even migrates, right? The bird is, who knows, right? So dosimetry for non-human biota can be complicated. And this complication, there are a lot of controversial papers in the literature that are simplifying the dosimetry and saying, oh, at these background level radiation dose rates, we're seeing mutations. So clearly radiation is worse than we thought. When actually that's not the case. Actually, there's a little bit weak dosimetry. So that's why dosimetry is so important, because we want an accurate dose-effect relationship. We don't want to be underprotective, but we also don't want to be way overprotective. That's not economically reasonable. So for the hydroponic uptake studies, 1 week of laboratory acclimation. So Nimisha, who you saw on the previous slide, the plant physiologist, germinated these plants for us in like a sand mixture. She brought them out. Dawn put them in these Erlenmeyer flasks. And you can see the nutrient solution here. Those Erlenmeyer flasks were then covered in foil to prevent light stress to the roots. 4 groups of 4 different plants because we had 3 different sampling times plus 1 control group. And a Hoagland nutrient solution is just a specific type of nutrient solution. There were 2 different experiments. You can see these plants, there's more green here. These plants are older. And then you just have a couple of little baby leaves here. These plants are younger because we were interested in the rate of uptake as well. The hypothesis being that a smaller plant is going to be growing faster and therefore might have a higher rate of uptake. 3 spiked groups, as I said, 1 control group, and this is what that looks like all set up. These plastic tubes are just air lines. There's basically a needle at the end of it bubbling some air into these Erlenmeyer flasks to oxygenate the roots. Harvest at 1, 3, and 5 days. We didn't go longer than 5 days because at that point the plants had sucked up a lot of the nutrient solution, and we didn't want to switch out the nutrient solution because that would change the composition. So these plants were able to go about 5 days before they needed new water. Separated the shoots, the green part of the plants, and the roots, and we took 1 group for autoradiography to get a broader view of how the radionuclide partition in the plant. They were dried at 50 degrees C to a constant mass. This is what those plants look like. Benchtop digested. This took forever. Technetium volatilizes at a fairly low temperature, so they had to digest on the bench instead of in the microwave. So I think this digestion took something like 3 weeks to do. And then analysis, either this is a liquid scintillation cocktail rack for technetium or for ICP mass spec. Autoradiography, probably the most interesting thing. Intensity correlated with growth time, and then we have the root-shoot connection or the shoot tips appeared to have higher content. I was saying plants have strategies for dealing with heavy metals, and we see that in this autoradiography. So at day 1, you see this pointer? Yeah. So this on the left-hand side, that's where the roots were connected to the shoots. They were connected here. And this bottom part is the root ball just in the bottom of the Erlenmeyer flask. Day 1, it looks maybe a little bit darker towards the edge, and we have some hot spots here. But then as we get to day 5, again, the left-hand side is where the roots connected to the shoots. We see definitely a higher concentration just before going into the shoots. And we see the plant also kind of moving that technetium out to the older part of the plants. It's pretty interesting. And then for the seedling plants, this just gave us a perspective of what we might see when we compare that. And then on the right-hand side, you see that we had a control just in case to make sure we weren't, seeing some kind of natural radioactivity here. But you can also see that the longer the plants are grown, of course, the higher technetium concentration there is. For technetium, this is the only energy appropriate for autoradiography, so we wouldn't see the other nuclides. And we actually use stable cesium instead of radiocesium for ALARA, or as low as reasonably achievable purposes. And we measured cesium on the ICP mass spec. So these are the results for technetium. The roots are in orange on the left, and the shoots are in green on the right. The plus signs are the seedlings, and the circles are the established. And so we do see that the concentration ratio for the seedlings is higher than that for the established. And we also see for the shoots that we have this increasing trend, super obvious in the seedlings, a little bit less obvious in the established, but it looks like we haven't gotten to equilibrium yet. Concentration ratios are applied at equilibrium, whether or not we're at equilibrium, generally in environmental protection. For cesium, similarly in the shoots, not much difference in the roots. Uranium, goodness, if we look at the x-axis for technetium and cesium, they're about the same. If we look at the y-axis, and if we look at the y-axis for uranium, wow, look at this, 10 to the 4. That's crazy. Uranium loves to sorb, right? So rather than going into the roots, what's going on here is that uranium is like stuck to the sides of the roots just because it wants to be sorbed to something. We still do manage to get some translocation into the shoots. And then neptunium is about midway between like the technetium, cesium, which both have nutrient analogs, and then the uranium. So a good bit of sorption in the roots, but then a increasing trend where the seedlings are higher than the established in the shoots. So in general, we see the partitioning is related to the different chemistry of these particular analytes. Concentration ratios vary. And roots generally have higher concentration ratios. This is important from a dosimetry perspective because concentration ratios are often used to figure out transport into the food chain, not necessarily to assess risk to the environment. And so risk to the plant, in this instance of uranium, again, these are fairly low levels, but hypothetically speaking, if you have a radionuclide concentrated on the roots and not in the shoots, you might be missing some potential effects there. So it's important to kind of think about that roots versus shoots distinction. And then plant age influences uptake, as we thought. Just a comparison to concentration ratios that are reported in the literature, shown in gray and in white. These aren't exactly comparable because these concentration ratios are for soil and not hydroponic, but just to kind of give you an idea. Uranium is certainly different because of the soil. Uranium is going to sorb strongly in soil and not be as inclined to go into the plant. But interestingly, there's hardly any information for neptunium. So this is a pretty useful study, particularly for neptunium. For the soil columns, this one's still in progress. It's been going on, for probably about 2 years, just because it takes a long time to do some of this work. Dawn designed these columns. We have a top port where she introduced the radionuclides, and then this bottom port where she sampled pore water, and then a collection cup for effluent at the bottom. 50-50 sand and Savannah River Site soil because the plants like that better and they grow better with the little balance of sand. We had 6 columns of the plant in a column with radionuclides, 6 controls where we had a plant without radionuclides, and then 6 where we had a column, no plant, and radionuclides. So we have a nice experimental setup to do an analysis of variance later on. Acclimated, and then after 4 weeks, this right-hand column, they were harvested, and then the columns segmented. And so you can see there's lots of roots in here, right? And so we're really wanting to get a profile for root concentration as well as soil concentration. So what happened was these columns, all 18, were segmented into 1 centimeter slices, and Scott, my undergraduate who I said I needed to give props to, dug out all the roots from all of these slices so that they could be measured and processed. That's a lot of work for him to go through that, a lot of meticulous work. Similarly, Hayden also helped with some of this, and he also did a lot of the weighing associated with this work as well. They were processed similarly to the hydroponic studies. Soil took a little more work. So this is the effluent. Blue or dark gray diamonds are plants. Orange circles are no plants, and then a triangle is the control. This is the effluent, so when she irrigated the plants and water came through the bottom, this is what was collected in that effluent. And we have effluent collection along the x-axis, and then we can see the concentration go up and then go down as we start having washed most of the technetium through the column. And we can see here, after day about 6, there's no more data, at least for the plants, because the plants are so thirsty, there's no effluent coming through the bottom. So the plants are taking up all of that water. But still, we can see the concentration of technetium being retarded through the column. For cesium, that's weird. Look at those yellow triangles. That's supposed to be the control. Everything's about the same. Turns out Savannah River Site has a very high, comparatively, concentration of cesium in it naturally. So there's not a lot of information we get here besides the fact that, wow, look at how much cesium is in Savannah River Site soil. Similarly for uranium. And then we got some good information for neptunium too that is very similar to the technetium. So that's for the effluent. We also have concentrations for the plant parts, both roots and shoots. Again, the cesium and uranium, the bottom graph, not super exciting because there's so much natural cesium and uranium in the soil, but for the technetium and neptunium, we do get a good bit more information. Notably, that technetium has higher translocation, and it probably would continue to go up if we continued this experiment out. Neptunium is fairly consistent between the roots that transition between roots and shoots, and then the above ground part of the plants. And then preliminary results for soil. I'm showing you the one that's not exciting first. What this does tell us, though, is this is the variability we can expect. So this is just one column for each line, and she has 6 columns, so we'll eventually get error bars on this data. But because there is this high natural concentration of cesium and uranium in the soil, this gives us a good idea of what we can expect the spread of the data to be once all 6 of those columns are processed for the more exciting data, which is the technetium and neptunium concentration in the soil column. So 0, this level 0, and then plus or minus 1 centimeter, because when you inject the radionuclide, it naturally spreads a bit. That's where the source is. So anything above that 0 line is migration upward, and anything down below that 0 line is downward migration. And so for the technetium, we definitely see that this, again, the dark gray diamonds are retarding the transport of cesium through the column. And we can see with the no plant that the technetium has effectively either made its way all out of the column, and what's remaining has diffused fairly evenly throughout the column. So that little bit of upward movement is due pretty much just to diffusion. For the neptunium is a little bit different. We see that this section of the roots seems to be holding on to that neptunium pretty well. And so it still hasn't completely made it through the column. And at the bottom here, we have kind of flip-flop what we see in technetium. And so the next step for this one is to, 1, look at the breakdown of the root concentrations, and also look at the masses and see if perhaps there's a bit higher density of the roots around where the source was that might be holding on to that neptunium. So as I said, this retardation of downward movement, one of the reasons for that is evapotranspiration and the movement of water upward and the plant evaporating that water out or transpiring, and the movement of ions through the column, and then also specifically plant uptake, or pulling the technetium in particular into the plant. Cesium and uranium, nothing super exciting, which brings us to the dose model development. This is my favorite part because I think it's fun. For these computational phantoms, anthropomorphic, technically, this would be like, I don't even know, like anthropomorphic would be like human-like, but since they're not human, I don't even know technically what that word would be, but they're incorporated into radiation transport codes to figure out where energy is deposited and then figure out dose rate. Primarily, this has been advanced in the medical side of things. So we started with the ICRU sphere. In my physics class when I was in college, my professor always said, assume a spherical cow, and that's what we did, except it was a person. We got a little bit better computing power in the 80s, and so we started making more realistic shapes, and a lot of times we can still use models like this. They're a nice conservative approach called a stylized phantom. We can take a three-dimensional image, say a CT or an MRI, and then we can translate that into a 3D image here and mark the different organs. These are called voxel phantoms, but these are rigid. They're not movable. And then finally, the fanciest one utilizes non-uniform rational B-spline surfaces. Sometimes they're called mesh phantoms. These are movable, so you can simulate a heartbeat. You can simulate breathing, walking, so they're pretty cool. And this is the model that the ICRP is going to for humans, the mesh models. So to give you a little bit of difference, this is a representation of a fish brain specifically. When you're figuring out your model, you have the organ from your CT and you have the different slices of the organ. That's 3D rendered. When it's voxelized, you see these stair steps. That's the rigid nature of just connecting the dots between the slices. You put that into a 3D CAD program, kind of like what Pixar uses in computer animation. You can smooth that out. And so this is actually what a fish brain looks like. So that's the difference between a voxel and then a hybrid model. ICRP currently just is everything is a sphere or an ellipsoid of some kind. So we're trying to do a little bit better than that. Some phantoms have been made before. A duck, a frog was the very first one for use in environmental protection, a dog, and there's a variety of others. This Arabidopsis and pine tree one, the pine tree hasn't been published yet. The Arabidopsis thaliana is just 2 ellipsoids instead of 1. It's better. So we tried a stylized model with 3 cylinders as our chutes for the stylized. That's really easy to make. It's quick to put and run in a computer. It takes maybe 30 minutes to run. For our fancier model, we have a micro CT, which is pretty cool. MI Labs engineered this, so we have a vertical gantry, which is pretty neat, so we can take x-rays of columns pretty easily. We marked, when I say we, actually Dawn's daughter did this, outlined all the different slices of this plant, and then it was 3D rendered into this voxel model. So a little bit better picture of that. So the voxel model is as you see it on the left. When we put it in Rhinoceros 3D, we smooth it out. Also notice in the CT on the top, some of the leaves got cut off, but when we put that into our CAD program, Dawn was able to add that back in by hand. So that's the benefit of this 3D program. The hybrid phantom, it's a hybrid between the mesh phantom and the voxel phantom, because in order for it to go in our radiation transport code, it has to be basically re-voxelized for MCMP to understand what's going on. So that's why it's a hybrid. So it still looks a lot nicer. You still have this top area, and you have a lot less discontinuities than the original voxel phantom. So just to give you a comparison, the output you get from this kind of code is usually a dose rate per activity concentration that you have in the source. So all this is showing us is 1, how our different models compared against each other, and 2, how they compared to the ICRP's ellipsoidal model. So that's what we're looking at here. So the purple dotted line at the top and then kind of towards the bottom on the right-hand side are 1-to-1. So you can see for neptunium and uranium, there's a pretty good match between all the models, and they're pretty close to what the ICRP ellipsoidal models are. For cesium and technetium, not as great. But also this is for internal exposure on the left, external exposure on the right. Because this is technically an aquatic system, the ICRP has both terrestrial and aquatic plants, but for the aquatic plants, they assume the plant's submerged. So what Don has done here is compared it to half of the aquatic. But still reasonable representation for the aquatic. So the aquatic is a better fit than the terrestrial here. This is just an example of how you could combine our dose coefficient or dose conversion factor with the uptake data or the concentration ratio of data we found in the hydroponic experiment. And just to give you a perspective of the amount of radioactivity we use, this derived consideration reference level, this is a point where you would probably want to intervene. So again, just kind of a comparison for the different time periods and how you would do that. What I think is interesting about this, though, is if we consider the contribution. So we have contribution from uptake, so radionuclides that have come into the plant and the positive energy. We also have external exposure from the hydroponic solution. And then if you're a root, you might be getting exposed from the shoot and vice versa. So the different contributions to the dose are shown in these pie charts along with the associated dose rate. So perhaps unsurprisingly, look at uranium. Almost its entire dose is from the roots, for uranium attached to the roots, which makes sense. Whereas cesium-137 has that 662 keV photon, so it has a broader distribution. Concentration ratios don't always take those things into consideration as they currently are. Different benefits and drawbacks. So the ellipsoidal models and the stylized models both are really easy. They're quick. My favorite, besides the fact that hybrid models are fun to make, the stylized seems to be the best bet for bang for your buck. It seemed to be a pretty good comparison to the voxel and hybrid model, pretty quick to make and pretty quick to run through the computer system. But if you have a hybrid models are cool to look at. So if you have a public-facing dosimetric model that looks most like the organism that you're trying to model and predict a dose for, there might be additional stakeholder buy-in from there. Whereas if you're like, oh, I'm going to model this, the angel oak tree, for example, in South Carolina, all these branches, all these roots, you know, we're just going to model it as a sphere, right? If I'm a stakeholder, I'm going to be like, I don't know that that's the best way to do that. So that's another potential benefit of these hybrid models. Also, once a hybrid model is made, it's a good bit of work to get there, but it's easy to deform. It's easy to like copy and paste, and we can make a whole field of this plant. We can make it bigger. We can make it smaller. So once you have one made, also anyone who makes this model wants to share it, wants people to use it. So once you get a database of these models, then it becomes more reasonable to use a hybrid. So using a hybrid that's already there is going to be much easier than trying to go through the process of making one of these hybrid models. So major findings, higher fidelity, hybrid models, a lot of flexibility, but still some limitations. It takes, we run these on the supercomputer, that transport code. Normally dose rates from activities that we're concerned with would be low, so conservative models are usually appropriate, but in the instance that you do exceed a screening value, these models could be used in a graded approach to gain additional realism to see if you really have passed a regulatory limit. And then it's also consistent with the international approach in getting more, or I should say higher fidelity models, more realistic models in this holistic approach of protection of human health and the environment. And that is it. Any questions?
So, questions? Ready, fan?
that's an area of research people like myself are really excited about and has been going on a long time. And so people still certainly do research in that area. And there's a couple of strategies. One, using plants to basically fix the contamination so it doesn't move off. There are other strategies where you're trying to extract, and then you take the plant and then burn it and deal with that or whatever. But to my knowledge, there hasn't been just a great use of that. At Savannah River Site, they do have constructed wetlands to help deal with heavy metals, and that has been pretty successful. So there are some certainly applications of using plants to manage contamination in the environment.
Yeah, so you mentioned that the grass that you guys used was fairly resistant to stressors like some unpredictable water or what have you.
South Carolina summer.
Is there any reason to suspect that infrequent watering or frequent watering would change the uptake response?
Yes. You want me to elaborate?
Yes, please.
Yeah, certainly. So for this, that day 6 or whatever, where the plant was clearly would have liked to had additional water, Dawn kept the watering regimen the same, so just to keep the method. But in other studies, for example, in the plant physiology department, they'll change their watering strategy to, rather than keep the water that's coming through the same, because that's what Dawn cares about. From the plant physiology standpoint, she wants the plant to basically have the same uptake rate throughout. So it certainly does, but trying to normalize that, we'll probably take a look at that for the data. But yes, it absolutely will change the uptake.
Thanks for the talk. I enjoyed it. I had a question maybe a bit orthogonal to your current work. In agronomy, they've used a lot of hyperspectral imaging methods to do things like measure nutrient uptake in vast fields of agriculture. I was wondering if you thought about something like that to look at translocation rates of the heavy metal radionuclides that you mentioned.
Yes, great question. I actually did some of that type of work in my dissertation. We were looking specifically at cesium and lithium from a chemical standpoint as opposed to radiological standpoint. And there has been some luck with that from like a higher scale, like a canopy scale look at things. But I'm not up to date on that literature to know where that currently stands.
Big push for phytoremediation at the time that Savannah River Site. And we did some phytoremediation. But there was even companies set up for phytoremediation. But basically, we invested a lot of money, we abandoned it because there was too much to control. The birds, the bunnies, and everything else flying away with it. And being able to control it. We actually did show that we could add nutrients and add surfactants to get the radionuclides to move up into the shoots at specific times and then harvest them. But even that, when we did the modeling of it, didn't look like it was going to be feasible. We also did some work for the Department of Energy and lead site, where they didn't have too many restrictions on us, so we were doing that. Has any of your work shown that we should revisit some of that?
It's an interesting question because I feel like phytoremediation is one of those things we just really want it to work. We really want it to be practical. But as you're saying, it seems like, at least in my literature review, that phytoremediation kind of has this circle. We get really excited about it and try it, and we're like, oh, we're disappointed. And then it kind of goes down, and then it comes back around maybe this time. Oh. So there's a current study going on right now in New Mexico looking at using phytoremediation in various uranium tailings areas. Haven't seen much come out of that, most of the work I've seen, modern phytoremediation, including my own, is yes, we can get plants to take stuff up, and that's cool, but then what? Like, the management aspect of phytoremediation to me seems is what's missing.
It takes a long time for you to basically get the soil down to very low levels by using phytoremediation too.
Yeah.
So it's better just to dig it up
Yeah. Or do something to try to leave it there.
Right.
Yeah. Yeah. Right.
When you say a long time, what's a long time? Is it like many years?
We modeled it out, and it would take 30 or 40 years to get down to very, very low levels.
Yeah, because like there was one, I forget. It was in the news when it happened, but there was like a float with sunflowers on it in like the Kiev Reservoir or something around Chernobyl. And everyone was real excited because sunflowers really suck up uranium. They're hyperaccumulators. Everyone's like, yes, look at all this uranium that went into the sunflowers. It's very exciting. There's still just a ton left, right? So as you're saying, it just is, you'd have to harvest and grow and harvest and grow. And yeah.
So are you plans to model the soil columns, soil plant columns?
Yes.
So I saw earlier in your presentation you didn't talk about it today, but you were using like a partitioning coefficient for the soil interaction.
Yes.
Given this discussion about how long it takes, do you think that non-equilibrium effects between the soil and the pore water are going to be important in that system?
Yeah, I think so. And probably what's going to happen is Dawn is going to take the early stab at modeling this. And then now we've seen like about how long we need to do this experiment to get to equilibrium. Probably the next student to come in might do something similar, revise it, and then go long term to try to get to equilibrium. But certainly it will. Eric? Oh, I guess he needs the mic.
Left-handed, so I'm not Patrick Mahomes.
Touchdown.
Excellent presentation. My question is, what dose, what magnitude of dose are we concerned with to the plant? Like, is it a gray? Is it a tenth of a gray? Or where are we getting concerned about there's a significant dose to a plant that we can start looking at what the risk is from that dose effect concern?
So the Department of Energy guidelines put that at about 10 milligray per day. So in general, non-human biota exposures are measured in dose rate because they're in it, right? So to do a cumulative dose isn't as useful in those contexts, but in terms of actual population damage, you're right, on the order of a gray. Maybe more because grasses are pretty resistant. The 10 milligray per day is like the management standard.
And that's a DOE limit?
Mm-hmm.
So in the past, the assumption was as long as we protect the human species, right, from any effluent or any cleanup activities, everything else was okay.
Yep.
And now we no longer believe that's true. So in addition to cleaning up, we're now
At least giving consideration to, it might be the case, right? That if we are adequately protecting humans, that we end up protecting the biota as well. But that just hasn't been shown. So the ICRP's goal is to take an approach that we haven't shown that yet. And if we can show that, great. Then we'll go back to that view. But for now, we don't know that for sure. And there are a lot of places where we have environments that we don't have people. Yeah.
Last question. Bioremediation has been used successfully at a number of sites that DOE has declared victory. Fernald is one of them. And so if those plants are getting more than that threshold, I guess they're grandfathered.
I think they don't care because it's a different, like you're not trying to protect the ecosystem, like you brought those grasses in specifically for a purpose, and those grasses are going to be sacrificed later anyway, right? Yeah.
Thank you very much.